Good afternoon, everybody. It's my great pleasure today to have two of the students working in our lab on a topic that I think, even though the title is about rare, it's not so rare if you take into account all the rare diseases, they are common. And I think in neuropsychiatric disorders, it is very common to find a patient with some peculiar phenotype and some difficult to diagnose syndrome. And in this case, genetics is very important to help us delineate the problem of the patient. And in some cases, this can be rewarding, except of ending the diagnostic odyssey of the patient. We can also sometimes benefit from some treatments that are specific for the patient in question. So I will not miss more time. I will not make any other delay on the introduction. I would like to present Dr. Minas Drakos. Minas is a PhD candidate in our lab. He's also a medical student. He has been working in the past few years on the diagnosis of rare neurogenetic disorders and rare neuropsychiatric disorders. And he's going to introduce us into this field of the rare disorders that somehow are of interest for many clinicians. Mina, we're happy to hear you.
Hello, thank you for having me. So I will share my screen. Tell me. Okay, so you can see it?
Yes. All right.
Okay.
Please go ahead.
Yes. So we live in a pivotal moment in medicine. So we transition from one size fit all approach to a more precise type of medicine. So we try to use data like the physiological status of the patient, his microbiome, his omic, like a genome, to tailor our treatment for better medical outcomes. Regarding the genome, we living a very important moment since the Human Genome Project, which it had a cost of the first genome sequencing about 3 billion. In 2001, in 2007, the cost declined to about lower than 1 million. With the NGS technology and its commercialization, we had the ability to sequence much cheaper our genome with less than $1,000. And today there are new platforms that can sequence the whole genome to about $80. So we have four main hot areas in the medical genomics field. We have cancer genomics, we have pharmacogenomics, prenatal and newborn genomics, and we have genomics for the diagnosis of rare genetic disorders. Regarding rare diseases, it impacts about 350 million people around the globe, 30 of them only in Europe. It is very important to understand that most of these disorders, about 80% of them, are of genetic etiology. Today, we have about 10,000 different distinct clinical rare diseases phenotypes, and it's very important also to have in mind that most of them affect children. Meanwhile, 50% of all these patients remain undiagnosed, and a very large percent of them have a misdiagnosis. Thus, we're talking for our diagnostic odyssey. The patient have to go in the healthcare system again and again, many times without the right diagnosis. So this increased the burden in not only in the healthcare system but also in the health of them, and also for the family. how many rare diseases do we have? As we said, we have more than 10,000 different types. Most of them fall in the neurological and developmental spectrum. Here we have how the increase of the distinct phenotypes. So you have OMIM, which catalogs the different Mendelian disorders, and you see that we have a large number of different phenotypes, and also a large number in genes that are implicated in these rare diseases. But how rare is rare? Most of them have a very low prevalence. So, for example, in a study of 3,500 different rare diseases, most of them, about 3,000 of them, have had a prevalence of lower than one in a million. So we have to consider that is how difficult it is to diagnose a patient that has a disease that is very rare, and how also difficult it is to characterize a new phenotype when you have a patient, for example, to 20 patients with a specific disease, and one is situated in, for example, in Greece, another is in the States. How can we bridge the gap and characterize this new phenotype? So these problems and these thoughts was considered in the EU and wanted to tackle them. So in 2018 to 2024, Solvar-D project was initiated in 2018, and the EU gave a grant of about 50 million euro to bridge this geographical distance, and also have closer the different clinicians and researchers of rare disease patients. And also another very important thing that this project done was to centralize and systematically analyze and reanalyze patients that remain undiagnosed. They pulled the genomic data and generally molecular data of patients that did a molecular testing but was negative, and through the analysis in a systematic approach, and also reanalyze them, they were able not only to diagnose them but also to find new gene phenotype associations. So Solvar-D gave the proof of concept that something was able to be done. And then came Erdera, which was essentially built upon the achievements of Solvar-D and other rare disease projects. Now this project that will started this year will end in 2031, has the aim not only to diagnose rare disease patients, but also to give the possibility to advocate about them, and also to find new treatment and fund basic clinical research. So this is where we are in the rare disease field in the EU. Now I will go you through the path that we do in diagnosing molecularly rare disease patients. It's very important before performing a molecular genetic test to consider some things. It's very important to see if we have a strong clinical suspicion of a genetic disorder. Does my patient has characteristics that will lead to having a diagnosis of a rare disease? It's very important to answer this. And also, does his phenotype fits to a specific genetic disorder? And if yes, how many genes are implicated or may lead to this disease? But what about the penetrance and expressivity? Is there any age dependency in his phenotype? And also, do we have any phenotypic overlap? Are there some different diseases that match phenotypically. This is very important also to have in mind. And the next thing that we have to do is to decide which genetic test to have. But yeah, I understand that many clinicians and have a problem deciding which is the best approach. How do we start, and which and with which molecular approach to continue. However, there are some criteria. For example, in the UK, they have this national genomic test dictionary where you can find the guidelines when to perform a genetic test, and not only when, and which genetic test you will use. For example, in early onset dementia, if you have an unexplained dementia in a patient and the age of onset is less than 55 years old, and you have excluded acquired causes, like for example a tumor or stroke, then you can proceed in genetic testing. Or for example, another case of unexplained dementia and in a patient that has a family history of also the same type of dementia. You can also proceed in genomic testing, and in this case you proceed with whole genome sequencing, or you proceed with STR testing, short tandem repeat testing. And just to remind you, in regarding Alzheimer's disease, we have many genes that are implicated in the pathogenesis of this type of dementia. However, the Mendelian types of them, in the familial types of them, are essentially three genes: APP, PSN1, PSN2. However, another gene, ApoE, and specifically the haplotype ApoE4, was recently found that and suggested that when found in homozygosity, it will be considered as a distinct genetic form of Alzheimer's disease. Essentially, what this means that this ApoE in the ApoE4 haplotype is transitioning to a Mendelian type of AD. However, more data I think are needed for this. So in our lab, and through the work of Labros Mathiudakis, a postdoc research of us, did research regarding the genetics of Alzheimer's disease and FTD, and essentially what he found in the population in Greece was that related to the controls, where this haplotype was found in 7.4% of the control. The AD patients and the mild cognitive impairment have a significant difference to 23.2 and 25% respectively. And after this, it is also very important to have in mind that after we decide which test to do to generate the molecular data, the process is basically the same, almost the same for each type. So we need a biological sample, usually blood. We extract the DNA. We molecular process them. For example, we do genotyping if you do an array, or DNA sequencing if you do, for example, whole exome, whole genome. You need to do a bioinformatic analysis and interpret the results. And after you interpret the results, you communicate the results to the clinician or the patient. Regarding the interpretation, you need a specific pipeline, so and also specific platforms. Among the platforms that we use is GPAP, a platform that was developed and extensively used in the SolveRT project, and also will be used in the RGERA project, where you essentially add the phenotype of the patient in the platform, and also with HPTOR terms, and also you add the molecular data, and then you use the platform gives you the possibility to analyze the data with different types of tools for that make it possible to reanalyze your undiagnosed data and give a specific result. So here is the dashboard. So here you can see the name of the gene, the effect, the in silico, and the predicted effect of the variant in the specific gene, and also you have also in silico predictions about the variant that we use for characterizing the different variant. After, however, you understand that when you proceed, for example, with sequencing whole exome or whole genome, you have a notion of variants, and it is very easy to be frustrated, and you need a way to somehow prioritize your analysis, essentially which variant to look first. For this, for example, we use there are many tools, but I will show you here Exomizer, which is a tool that was built in neural networks. That what that does it done? For example, you have this variant. It scores the variant based on its predicted pathogenicity or and its allelic frequency. It gives it a score, a variant score, and also based on what phenotype characteristics have you submitted to the platform. It sees how does the phenotype of your patient matches the phenotype of the disease that this gene leads. If you have a large similarity between the phenotype of the patient and the phenotype of the disease that the gene leads, you have also a high phenotypic score. And then you have essentially a meta score, an exomizer score that essentially what says, okay, I have a high probability that this may be your variant. Another thing that that you should have in mind that most of genes that we have are in our genome. We don't know exactly what they do. So we sometimes have pathogenic variants in genes that they have characteristic of pathogenicity, but we don't know exactly what they do. So, for example, if we don't know what this gene does in humans, if we have information about what this gene does, for example, in a model, for example, in mouse or zebrafish, etc., it can also give you a phenotypic score. But for in this case, it specifies you that okay, for we have similarity of your patient with the phenotype that it does to the model. Very important as well. And even if you find another thing that we have to consider is to classify your variants regarding its pathogenicity. In other words, to see if your variant has many data to be classified as pathogenic. So we use and all clinical genomic labs must use the guidelines of the ACMG and AMP 2015 guidelines. That essentially what it does, it classifies the different variants as benign or as pathogenic. They use 28 different criteria, and you can classify your variant from benign VUS, which is a variant of unknown significance, or pathogenic. When you have a VUS, what essentially this means is that you have contradictory criteria. So one criteria is, for example, of benign, another is pathogenic, and you have a conflict, and then essentially your variant resides in this large region of VUS. However, to report a variant and to have a clinical decision making or on this report, your variant must be at least likely pathogenic. In other words, 90% probability that this variant has pathogenic characteristics. And also another thing to keep in mind is that these guidelines are used for SNVs and small insertion deletions, and for Mendelian disorders, for polygenic diseases or oligogenic like retina pigmentosa, you cannot use these guidelines. And also you can use it for an familial cancer, but not for somatic cases. Moving ahead, it is very important also to see and assess the genome phenotype fitness. How much does it fits this gene that you find in the phenotype of your patient? Because if we sequence our genome, your genome, I will be sure that I will find pathogenic variant, but this does not mean that you have a disease or that the disease that a patient has matches this pathogenic variant. So you can use the different catalogs that you use about different diseases. Here you can see OMIM or Orphanet, which has the clinical presentation of the different disorders that each monogenic of the different phenotype that each gene that has. And also after that, it's very also important to report back genes that to be sure that the genes that you're looking are curated. So in the literature, you will find many times genes that have been implicated. For example, one paper to implicate it with a specific disorder. However, there's not. If you search more, you will not find a second paper. So it is very important to look and report only genes that have been validated, essentially curated, that leads that we are sure that it leads to a disease. For example, here we have TTN gene that you can see it has been it has been implicated, for example, in the literature for dilated cardiomyopathy, and arrhythmogenic right ventricular cardiomyopathy. Here you can see that the classification they have curated, so the working group have sat down and saw that, okay, we have definite the evidence that for dilated cardiomyopathy, TTN will lead to this disease. And for example, for another type of cardiomyopathy, we have limited. What this means is that, for example, for the limited case, you cannot report this variant more than a VUS, which means you cannot report it and use TTN for arrhythmogenic right ventricular cardiomyopathy and do clinical decisions about this type of disease. For moderate disorders, you can report it as likely pathogenic, and for definite classification, you can go upper to classify them as pathogenic as well. So different type of evidence give us a different type of how much we can report it in the pathogenicity scale. And also they ClinGen also does not only curate the different genes but also curate the different variants. For example, if different variants are reported in the literature, the working groups curate the different variants as pathogenic or as benign. So for example, in this gene RIR1, they curated 14 variants that are reported in the literature as benign. So there are some cases, for example, in the literature that they were reported as pathogenic, but essentially they're not pathogenic, they're benign. It is very important to see this as well. It is also very important to assess the mechanism of disease. How does if this variant that you found leads to a mechanism of pathogenicity that is similar to the known mechanism of pathogenicity of the genes that you are looking. For example, if you find in the DNA level a missense variant in a gene that, for example, HTTT that leads to Huntington, that we know that it leads um, to to disease through triplet expansion, and you find a missense variant, this could not be does not fit. Okay, so it is a very common mistake that trainees do, that they find um, a variant in a gene that matches the phenotype, but they do not assess the mechanism of disease. Okay, so it is very important. It is not only the type of the variant. Sometimes it is the location, also the location. So for example, the triple expansion must be located in a specific region to be sure that it fits the mechanism. Okay. And also this was in the DNA level. In the protein level, we also have to assess the mechanism. For example, if you have a variant that leads to a truncation of your protein, which is predicted to lead to a disease through a loss of function, and and it is a gene that we know that it leads to a disease through gain of function. This is a mismatch as well. Okay, we cannot exclude the possibility that that this is an exception, but it's very important to also assess and match the variant matches the mechanism of disease as well. Another level of assessment also is the transcription level. This is also very important. So, for example, a gene has many transcripts. Okay, but it is very important to see if the variant it is in a transcript that is expressed in a tissue that we are interested. Okay, so for example, if we have a gene, if you have, for example, a transcript that is not expressed in an epilepsy patient in the brain, but it leads to a different type of disease, it's a problem. So we have to assess this as well. And finally, also to see the inheritance pattern. It is also to do essentially segregation analysis and see if the type of inheritance pattern also it fits it fits as well. We have also to keep in mind that we have cases of mosaicism. Okay, so not all cases are very are simple. Okay, and also we have cases of genes that are of genomic imprinting, which is also important to have in consideration. Even though that you have done all this analysis, you have many cases that are unsolved. For example, when you do a whole exome sequencing, usually the diagnostic yield is about 25, max 30%. Okay, so something is going on, something limits our ability to diagnose our patients. One thing is that obviously that we have a limitation of the technique. Okay, we have an increase about 10% if we transition from a whole exome to a whole genome. Another about 10% if we do an RNA-seq. And also not only the technical limitations, but also sometimes we don't know, for example, the gene that is implicated. We don't know the disease. For example, if you do a reanalysis of your data in about 2 years after it was negative, it has been shown that you have an increase in your diagnostic yield of about 10%. This is also very important to have in mind. That's why in our lab we do extensive reanalysis of undiagnosed cases. This is a poster from our work in the lab that was in the European um, Academy of Neurology this year, where we extensively reanalyzed 243 negative undiagnosed cases and patients, and we have a diagnostic yield about 11.9%. So it's very important also to have in mind that even we have negative molecular data, we have to reanalyze our data some years after the first analysis. Sometimes we have new genes that come to the stage. So for in the Erdera and Solvar-D, it gives you the possibility to do cohort analysis, which means I create cohorts. We can create cohorts of patients that have similar phenotypic characteristics, and then we align their genomes. And through this alignment, we are able to find new gene disease associations. This gives us the possibility to find new diseases from and come and describe the phenotypic characteristics of patients that are not in a close proximity. This is very important. Okay. However, this is very time consuming, so it is very important to find faster ways to do this patient matchmaking. So we in the lab try uh, to also make faster this. So HIAAD Samtsea also tries to through using neural networks to find better matches and faster in around the world patients. And finally, our aim through the Erdera project is to until 2028 to provide to this ecosystem more than 1,000 genomic data, give the possibility to reanalyze genomic data of undiagnosed patients free of charge, and also to sequence new cases, and also finally to create a centralized genomic pool of undiagnosed patients in Greece and in Crete and in Greece generally. Thank you.
So thank you, Mina, for this introduction. I think it sets the stage to present the data from Lina later on. I don't know if there's any question from the audience, or we should proceed to the presentation from Lina and see later on if there are any questions. I think maybe we should better do it.
Yeah.
Okay, so Lina Zapri is a master's student in our lab. She's working for quite a few time in our lab on the reanalysis of genomic data. After Mina set the stage by introducing rare diseases and how we approach the genetic analysis and reanalysis of these patients, Lina is going to present us the results of our lab and what's our experience with the analysis of patients that have been extensively studied in the past and still we have not reached a diagnosis. So Lina, please proceed.
Thank you for the introduction. My name is Lina Zapri, and today I will present to you the analysis results of 243 genomes from a genetic database of neurological and other diseases. So I want to start with mentioning some examples of the first round genetic analysis in dementia patients that we conducted in the lab. The first example is a cohort of 130 patients with FTD, ALS, and both FTD and ALS. So among these patients, 14 of them had a whole exome sequencing performed, and one of them had one targeted gene sequencing as initial test. The rest of them, the 115 patients, had a C9 or 72 repeat expansion analysis as initial test, and 14 of those were found to have a presence of this expansion. For the rest 101, we found no repeat expansion, and thus we performed the whole exome sequencing. Screening the genome of those 130 patients, we found that 25 of those patients carried pathogenic variants in the C9 or 72, GRN, VCP, DBP, FUS, and SOD1 genes. Specifically, 14 of those patients had a pathogenic C9 or 72 repeat expansion variants. 3 of those patients had pathogenic variants in TAR-DP. 3 of those had pathogenic variants in GRN. 3 of those had pathogenic variants in VCP. 1 of them had one pathogenic variant in FUS gene, and finally, 1 of them had pathogenic variant in the SOD1 gene. So in this study, we found that a male who was 60 years old and was affected by FTD and ALS had an isoleucine to valine substitution at the position 383 in the DBP protein. The same variant was identified in a patient, in a female patient who had ALS. And what is really interesting here is that we conducted a separate study, and we found that 2 patients who exhibited symptoms of Alzheimer's disease carry the exact same variant with the rest of the 2 patients. So this is not out of surprise because we know from the literature that 2 different types of dementia, and generally different types of dementia, can be caused by the exact same gene. This is a second example of a whole exome sequencing diagnosis in a 65-year-old female patient who had cerebrovascular events and dementia. Specifically, this woman presented with headache, depression, 4 strokes, and dementia. Her son also exhibited disease-like symptoms, and he presented with headache, stress disorder, and sleep difficulties. We did a whole exome sequencing analysis, and this analysis revealed the presence of one pathogenic NOTCH3 variant in the female patient, and also her son, who is affected by the exact same disease, also carries the pathogenic variant. The 2 patients have a variant within the EGFR domain, which is a domain that contains many cysteines, cysteine residues, and this domain is typically affected in CADASIL patients. The variant carried by both patients alters the 3D dimensional protein structure, and thus the NOTCH3 protein leads to a disease-related phenotype because it dysfunctions. A third example of first-round genomic analysis was that of an 18-year-old female who was affected by visual phenomena, myoclonic epilepsy, and dementia. Until the age of 14, she was healthy, and she presented the symptoms afterwards. A whole exome sequencing analysis revealed one pathogenic variant of EPM2A gene, which was in homozygous state. So the patient was homozygous for this specific variant. This gene codes for the protein laforin, which plays a key role in glycogen metabolism. And this gene is associated with myoclonic epilepsy of LaFora1. To prove that the variant that we identified was indeed responsible for the female's phenotype, an axilla biopsy was performed, and what we observed is that there were glycogen bodies, which resulted from the dysfunction of laforin. So despite the molecular diagnosis of some patients, some other patients remained undiagnosed. For those patients that remained undiagnosed, we conducted a further reanalysis project in which we reanalyzed 243 genomes of patients with rare diseases. Specifically, those patients were of Caucasian descent and were living in Greece, mostly in Crete. The patients had heterogeneous clinical presentation, and their age ranged from 0 to 78 years, with a median age of 15 years. In all cases, there was a suspicion of a rare disease, and this suspicion led to a whole exome sequencing performance using DNA extracted from whole blood. The majority of the patients we reanalyzed were affected by neurological disorders, attributing for 70.8% of the total cases we reanalyzed. These were followed by developmental abnormalities, metabolic disorders, kidney diseases, and also other types of diseases were also observed. Within the neurological group, the majority of the patients we reanalyzed were affected by ALS-FTD, accounting for 46.5% of the neurological cases. Smaller numbers presented with myopathy, epilepsy, dystonia, and ataxia, and of course other neurological diseases. The patients affected by non-FTD dementia represented around 1.7% of the neurological cases. And so for this project, we reanalyzed a total of 243 patients from the Neurogenetics laboratory database. These patients had been analyzed in the past. However, the genetic cause of their disease was not identified. And for this reanalysis, we used the GPAP platform, which is an online tool that is used for diagnosis and discovery. This platform has implemented bioinformatics tools such as the exomizer that help variant prioritization. And also, we did afterwards the variant classification using the ACMG-AMP guidelines. We then classified the patients into three main categories. The first one is the likely diagnosis group. In this group includes patients in whom we have identified the variant, which explains their phenotype. However, confirmation through Sanger sequencing or RNA-seq is still pending for standard reasons. The second group is the uncertain diagnosis group, which includes patients in whom we have identified a variant that might correlate with their clinical picture. However, additional data is needed in order for this group to consider a likely diagnosis. We are optimistic that in the future, some of these patients will receive an accurate molecular diagnosis. And finally, there is the no diagnosis group, which includes patients that remain molecularly undiagnosed and that for whom we have found no plausible variants that explain their phenotype. So moving on to the results, we have achieved for the reanalysis project, we achieved a diagnostic yield of 11.9%, meaning that 29 patients out of the 243 patients that were analyzed, so 29 patients were diagnosed, and they belong to the likely diagnosis group. For the 7.8% of the patients we reanalyzed, we have found a variant that might correlate with their clinical picture. However, more data is needed, and this percentage of patients falls into the uncertain diagnosis group. So for the rest of the patients, the 80.3% of the patients we reanalyzed, we found no plausible variants. So these are the patients that received no molecular diagnosis. So the missense variants accounted for the majority of disease-causing variants in patients from the likely diagnosis group, and these were followed by frame shift, nonsense, splicing, and other types of variants. The majority of patients diagnosed after the reanalysis were heterozygous, while some of them were homozygous, compound heterozygous, and hemizygous. For this project, we identified 14 novel variants, meaning that we found 14 variants that had not been previously described or published. And you can see some of them in this table, and we are now in the process of submitting those variants into the in the ClinVar database. So this is a real case. There was a 22 female who was affected by autism, hypotonia, developmental disorders, and immune deficiency. And this patient performed a whole exome sequencing analysis. In the first analysis, there was we identified the HIAAD4 gene, which is likely pathogenic. However, it does not fit the phenotype. So this is why we decided to do a reanalysis in this patient. We found a POC-Z variant, which is likely pathogenic, and it is actually a frame shift stop-gained variant. This variant is associated with the White Sutton syndrome, which is an extremely rare syndrome with only 150 known cases up to date. It is inherited in an autosomal dominant manner. However, most of the cases are because of de novo mutations. The age of onset is a very young age, and this syndrome is characterized by cognitive decline, developmental and behavioral problems consistent with our patient's phenotype. This is a second case. There was a 9-year-old male who was affected by developmental disorders and skeletal dysplasia, among others. On the first whole exome sequencing analysis, there was no causative variant found. On the second whole exome sequencing analysis, we identified the PUF60 variant, which was likely pathogenic and was stop-gained variant. And this, the PUF gene is associated with Verhey syndromes, which is an ultra-rare neurological syndrome. Only 56 patients have been described up to date, and it is inherited in an autosomal dominant way. The age of onset is again at a very young age, and this syndrome is consistent with our patient's phenotype, as it is characterized by developmental disorders and skeletal abnormalities. Here we can see some of the variants that we identified in FTD-ALS patients. And the key takeaways is that genomic medicine has and will revolutionize medical care. We reanalyzed the heterogeneous cohort of 243 patients using the GPAP platform, and we achieved to diagnose 29 patients out of the 243, meaning that we have a diagnostic yield of 11.9%. For the 7.8% of the patients we reanalyzed, we have found a variant that might correlate to their picture. However, we need more data. We have managed to find 14 novel variants and to diagnose patients with ultra-rare diseases such as the Verhey syndrome and the White Sutton syndrome. And also genomic analysis, genomic data analysis assists with molecular diagnosis of the rare cases of dementia. I would like to thank everyone from the lab, and thank you for the attention.
Thank you very much, Lina. Thank you for presenting us this data. I don't know if there is any question from the audience.
Indeed, yes, but
Yanni, go ahead.
okay. Thanks again for this great presentation. Both of you, really nice work. And I think it's a very good example that for us that we are more in the basic research of how using techniques like the whole genome analysis, you can not only identify new genes and give some tools back to the basic research, but also you can help diagnose our patients in Greece and abroad. And I find quite interesting the whole pipeline you have, Ioannis, you have built in the lab in Crete, and I think it will give a lot of opportunities for us. Question: do you publish this? So can we have the sequence for some of the researchers that they need to use them and try to study it as new cases, or new genes, or new combinations?
So, Mina, do you want to answer, or Lina?
So can you do the question again? So the question is if the gene that we have
If we have
discovered.
published, if we have published these new reanalysis cases.
So regarding the reanalysis, we want to collect more undiagnosed cases before publishing them. So we have in the first wave of analysis, we analyzed, as you saw, the 243. We collect currently more data, and we want to also try to compare different cohorts. For example, the neurological with the kidney cases, and so we have to harmonize kind of the data before publishing them. And however, we are planning to publish it. Regarding the new gene phenotype associations, we have found some cases. We have patients that have been matched around the globe. but we are trying to find more. We have done some functional data, not in the lab ours, but we have functional data that support, for example, the clinical presentation of the patients. But trying to find more patients around the globe. It's very difficult.
That's
For
for sure.
example, for example, we have a case that we have only three patients. I'm searching everywhere about more cases, but I cannot find. So we have to find more cases to have a solid foundation and solid, for being sure that this new gene leads to the disease. But they are in the way of being published.
So thank you. Next, Dr. Liketos, you want to ask something, and then it's Dr. Lazaros.
Thank you. Yeah, very nice work. You're all to be congratulated. I'm trying to think of ways to think about how these findings become therapeutics. What are the kinds of models that you're thinking of using to understand better the functional changes that occur with these mutations? And are you thinking at all of setting up models of that kind so that you can test repurposing of existing therapeutics?
So, Lina, do you want to elaborate on this, or?
Yes, of course. Well, identified the genetic cause of a disease is like the first step for the diagnosis is the first step. So next comes the therapeutics, and of course, I have recently read about the CRISPR on Down syndrome. Where?
Okay.
Oh.
So there are ways, possibly. I don't know, Mina, if you want also to comment on this.
Yeah. So also it is the next step. We're not much close. However, for example, creating models, it is very important. It is one of the goal of Erdera to go back to basic science and to be able to find ways to treat, firstly, biomarkers, okay, and also maybe because some of them may be treated if you are able to identify them very fast. However, we're heading to treating them molecularly. For example, as you know, FTD has gave permission for the use of CRISPR for silk cell disease. Okay, and also some are of them also in TTR amyloidosis. They are in the third clinical in the third phase of clinical trials. But I'm not sure if we are very close for the next years. I think in 20, 30 years we'll be able to treat them molecularly.
Yeah, let me
Yes.
put a thought into your mind, if I could, very briefly. So if for all these individuals you can obtain blood cells, PBMCs, transform them to stem cells, and then differentiate the stem cells into brain entities, I'll put it broadly, that you think are impacted by the pathogenic mutation, you now have an individual model for these rare diseases that is specific to the individual patient, whereby you can actually study the functional changes that you're seeing, whether it be neurons or whatever your other cells are. The advantage of creating that is that there is a vast library of well understood drugs that are on the market all over the world. And you can start thinking of ways to bring them into your models as a repurpose project to see if they will modify the functional impairments that you're finding. It's a big job, but I think in terms of these rare diseases, because they're not going to get much attention otherwise, that might be a quicker way to getting some therapeutics, because the development of disease-specific therapeutics, there's no incentive for these rare diseases to have that happen out of pharma. So just food for thought.
Yeah,
Yes.
sure.
Thank you for this comment. Yes, I just want to add that these projects, like the DERA project, have drawn attention because of this rarity. So there is not much interest to develop therapeutics for a disease that's rare and affects a few hundred or a few thousand people around the globe. Of course, if you develop a therapeutic, then this is also the price is different, and maybe some companies are interested. But through collaborative projects like this one, you can, have someone to do the genetic diagnosis and the clinical characterization, and maybe somewhere else there's a lab that works on a model that he or this lab can apply a possible therapeutic, let's say, agent, and of course this collaboration can move the field forward. And this is a nice,
And do you have access to PBMCs? Do you have access to PBMCs for
Yes,
these?
this is a very good comment, and we're trying to establish with Yannis Haralabopoulos. He's here, and also with Mimika Thomaidou from the Pasteur Institute. We're trying to establish collecting PBMCs from patients with Alzheimer's disease, and the next project is to establish PBMCs from patients with rare disorders. And when I mean rare disorders, for example, the patients with TDP-43 variants that have Alzheimer's phenotype or have FTD or ALS phenotype.
That's not
Because
so rare.
what
Yeah.
we
Not so rare.
sorry.
That's not so rare. TDP-43 is late is pretty common.
Yes, the pathology is common, but patients that develop Alzheimer's phenotype due to variants, or we think due to variants in the TDP-43 gene, are very rare. In our database, we found only 2 patients with Alzheimer's phenotype and variants in the TAR-DBP gene that encodes for TDP-43. So we have a big bucket, that's the Alzheimer's patients or the TDP-43, let's say, pathology patients, but only some of them have variants in genes like the TDP-43 gene. So maybe we can isolate these patients and target therapies for them. So this is an option for the future. Okay. So thank you very much. Because time is running out, Dr. Lazarus wants to ask something.
Okay, can you hear me?
Yes.
Okay, so thank you very much for your presentations. Both were actually very, very nice. So I actually wanted to ask a question about non-curated genes or variants that might be either non-curated or partially curated. So if you find such a gene in your pipeline, how do you proceed with it? I mean, do you discard it completely? Do you do something further in terms of analysis in order to perhaps keep it for further use?
Yeah, this is a good question, because we have a large genes that are not curated. So this is a process. That it's keep going. So and also because science is dynamic, so you have a paper that has strong indications that what it mentions. It is the disease that it shows in the data. We do not discard it. Okay, we keep it. You can report it. Okay, you report it, but you have to in the report say that this is an ongoing investigation. Okay, but for example, the question is, when you describe a new clinical entity, okay, you have to you will report it with the patient, okay, but because the curation will be done in 5-10 years when they have data, okay, because when you describe it, the disease will be the first paper. So you do not discard it, you can report it, but you have to keep in mind that there's a possibility of some mistakes when you do the report.
Okay, thank you very much.
Yep.
Thank you for the question. I don't see any other questions, so I would like to thank Minas and Lina for being here and presenting this work. I would like to also thank Dr. Sotiropoulos and Dr. Lamos and Dr. Liketo and everybody from HIAAD for giving us the opportunity to present this data, and to thank everybody for being here. I don't know, Ioannis Sotiropoulos, if you want to close, because you need to close the bridge.
Have a nice summer holiday, and we will see again with the next series of bridges of the HIAAD series on September. Thanks, and have a nice holiday on summer holidays. Yes.
Bye to everybody.
Bye.
Bye.
Thank you. Bye.